Welcome back as we wrap up Chapter 8 and the idea of cell transport. So we left off yesterday discussing passive transport, which ultimately requires no cell energy, no ATP, and it's going to go from high to low. So when we talk about diffusion today, we're going to see that diffusion, sometimes called simple diffusion, is going to be the one that biologically occurs across the phospholipid bilayer. So to keep things simple, I'll just write that PBL. And so this is going to occur with those substances we said that naturally can pass through. So CO2, oxygen, steroids, things that are very small and polar, or things that are nonpolar and still relatively small, will be able to pass through this way. You'll also see that we can apply diffusion to anything. That's like perfume, you could spray cologne, you could be cooking food, you can drop food coloring in water, and you'll see that they'll start to move from high to low concentration and spread out. So that's why you're able to smell it, but it takes time because this is not the fastest process. So that's why regular diffusion is not usually a very good thing if you're trying to live and you're large. It's not very efficient. If you're very small, very thin, it's not as big of a deal because you're not covering as much distance. But when you're looking at like humans, if we relied upon diffusion to get oxygen throughout our bodies, we would pretty much, I'm sure, die. So we've got to make sure that we've got methods of kind of improving this. In our case, it's making sure we have blood vessels that are right near the surface of our lungs and lots of tissue in our lungs, lots of surface area to make sure that oxygen can get into the blood and then the blood can swiftly be pumped throughout our body to get it where it needs to go. So this process is fine over short distances, but you really want to watch it over long ones. It's just not that fast. Now, osmosis and facilitated diffusion are just going to be other examples of diffusion. They're just special cases. Osmosis, as you'll see in a second, is just a special case of diffusion of water, whereas facilitated diffusion is going to be a special case where we allow diffusion to occur by using a protein that's typically embedded through the cell membrane, so an integral or transmembrane protein. So moving on here, we're going to cover osmosis. Now in osmosis, you got to pay attention to who we're talking about. So in this case, we're going to get a hypertonic solution. And hyper is going to be more. So we're talking about a solution here that has red blood cells that has more solute, more stuff in it than the actual red blood cells. And the way this works is the more solute you have, so in this case we've got more solute, that means ultimately the less water we have because that solute can bind to and interact with the water and essentially remove it from our pool of usable water in the cell. And so what that means is that the cells themselves, which would be considered hypotonic, meaning they have less solute than what's around them, have more water. And so because they have more water than the surroundings, there's a gradient, and so water wants to leave. The other way you can think of this is the phrase solutes suck. And so when you look at this phrase, solutes suck, what it's trying to get across is that whoever has more solute has less water. And because diffusion of water is going to be down the gradient, in other words, from where there's more water to where there's less, the water is always going to go towards the guy with more solute. It's always going to go towards the hypertonic guy. And so in this case, the solution that the cells are in is hypertonic. So these cells are losing water and they're undergoing what would be called plasmolysis or crenation, depending on where they're at. But it's basically this idea of shriveling, and that can kill a cell pretty easily. Then we've got isotonic, which for animal cells is what we want. And this one means that they have equal concentrations. So this is where there's really no appreciable difference between the amount of water or solutes. So you have some water leaving, some water entering, but the net overall shift in water is zero. Versus over here, you have very little water entering and a lot of water that's leaving if you're in a hypertonic solution. And then lastly, you have hypotonic, which means less. Hypothermia, low temperature, hyperthermia, fever. So hypotonic means that this one has less solute, and so that means that the cell must have more. And that means the cell's hypertonic. And so because we know water goes towards the hypertonic guy, solutes suck, the water is going to go mostly into the cell. A little bit will come out, but most goes in, and that means that the cell is going to swell. Now this may or may not be an issue. If you don't have a cell wall that's rigid to prevent you from bursting, it can in some cases allow for cells to rupture. So if you're in fresh water for a long time, you'll see that your skin can start to get wrinkly, and in some cases you can get this white film that forms that peels off because you've damaged a thin layer of outer cells, and they can actually rupture and die. You wouldn't get this as much or at all if you're in like salt water, because that one's more isotonic, so it's not going to have as much of an osmotic effect upon you. So depending on who you're talking about, you'll see that plants and such, we'll mention later, but they tend to like to be in a hypotonic solution. So they like to be hypertonic themselves, hypertonic cells. Whereas in animals, things without cell walls, they technically prefer isotonic. I'm not aware of a whole lot of stuff that likes to be hypotonic. I can't think of anything, quite frankly, off the top of my head. I know there are a couple things that are hypotonic that would be like living in the Dead Sea. I believe that those things would likely be hypotonic, but I'd have to look up to make sure if those archaeans are or not. So in general, it's going to be isotonic, or it's going to be hypertonic that the cells want to be. That's the norm. Whoops. So when we talk about osmoregulation, just to kind of quick give us a scenario, if I draw a beaker here, and I put a cell inside, and I use dots to represent the solute, if we're talking about a hypertonic cell, that means that cell better have more solute than what's around it. And the key thing here, and you'll see in the next slide, which I'll skip largely through, is frame of reference. Because the cell here is hypertonic, but if you have something that's hypertonic, there has to be something else that's hypotonic. It's one of those things like if you were rich compared to other people, somebody else has to be poor. Otherwise, everything's just equal. And so in this case, the solution then must be hypotonic. And so you really got to be careful when you look at this to make sure who we're talking about. So in this case, the cell is hypertonic, so water is going to want to go in mostly. A little bit will come out, but overall, there's an imbalance. So this one will swell. And if it swells too much, you can get lysis where it explodes, or, well, ruptures. So you just got to pay attention to frame of reference. If we have a hypotonic cell, if we kind of stay with talking about the cell, we'll see that that cell is going to have very little in the way of solute, but the outside is going to have a lot. Forgive my weird lines as I draw lots and lots of dots. And so because the outside has so much more solute represented by these dots, you can see that solutes suck. You know, outside ultimately is going to be hypertonic, and so you're going to get water that's going to leave the cell to go out there. A little bit will go in, but very little. So most of it's going to be going out. And then isotonic, this will be balanced, so if you see it drawn, just using the whole solute idea, I'll use some less dots here so it's faster, you should see where there's roughly the same ratio. So if you look at a specific region on your sheet that's a centimeter square, you should see about the same number of dots. And so that means that water will move. Water will go in, water will leave, but it's going to do so in equal amounts. And so in this case, we're going to get isotonic where it's equal, and we're going to see no overall change. Because there's lots of fun ways to detect this. For instance, you can mass the cells afterwards to figure out if they've gained water, gained mass, or if they've lost it. You can also measure them, and you could do like diameter, and you could see have they gained or have they lost, or just in general any other way to calculate volume. So there's lots of ways that we can kind of verify this, especially if you have a big enough sample. Obviously one cell is pretty tough to measure or deal with, but if you have a larger sample, you can analyze, is it gaining, is it losing mass, is it ultimately gaining or losing volume, to try to figure out exactly which scenario it's in. I've already said watch reference point, and then the last part here that I want to deal with is just in case you see water potential. Water potential is something that's usually drawn with a Greek psi, which looks like a little Greek trident that I just did horribly here. But the goal here is it's going to be measured in MPa, megapascals, or bars. I'm not going to freak out about units. I just want you to get the idea in case someone uses it. And the idea here is that distilled pure water is going to be essentially 0 bars or 0 MPa, a value of 0. And then as we add solute to it, it's going to become negative. So it's very normal when you talk about water potentials to have negative values, like negative 0.3. We can have this be MPa, megapascals, and we could have one that's negative 1.3 megapascals. And the goal here is that water always wants to go to the lowest water potential. That's the guy with the most solute, assuming that nothing else is involved. You'll see when we talk a little bit about the potato lab, we'll bring up a bit that there can be stuff you do. Like in some cases they can have these like little U-shaped tubes. It's not U-tube like the site, with a membrane in the middle. And so you can mess around with the solute concentrations to move water, but in some cases they can also add like a plunger, and they can physically apply pressure by pushing down on the plunger or by pulling up. And so there are other things that you can do to mess with this. But for our purposes, for pretty much the course, you just need to know that the more negative it is, that's where water wants to go. So negative 1.3 is the hypertonic guy, and that is where the water is going to move. So the 0.3 MPa solution will lose mass, will lose volume, will lose water, and the negative 1.3 megapascal solution will ultimately, or whatever it is, solution, cell, is going to gain that. So it would swell, it would gain mass, it would gain in volume. Now, plants and things that have cell walls, like many bacteria, are kind of a unique scenario where especially plants, they want to be pressurized. That's what makes them rigid, especially green plants. They work by being what's called turgid, and that just means pressurized. So the cell wants desperately to be hypertonic. They want to be in a hypotonic solution. And so that means that water is constantly trying to come in, which means the cell is trying to expand. Now, if this is an animal cell, it would rupture. You'd go through lysis. It'd blow up, for lack of a better way. Whereas in a plant cell, because of this rigid cell wall, it's not a problem. It just makes them pressurized, which is very useful for them because that makes them more rigid. If you put them in a solution that's isotonic, what's going to happen now is you've got equal water movement, so it's not really pressurized. And so that's what happens a lot of times when plants wilt, when they start to droop. And that's why people will say, oh, it's wilting, make sure you water it. Because you're trying to add more fresh water, close to pure water, to the solution, which makes the cell, which should then be hypertonic compared to that, be able to take in water. And so that will allow it to pressurize and become turgid. And then the worst thing for a plant that will kill it very quickly is if you put it in a solution that's hypertonic. This is what happens if you're Romans and you salt the earth. You put so much solute then in the actual ground, so the plant cells are now hypotonic and they lose water. And this causes plasmolysis, where the cell membrane actually pulls away from the cell wall, and it makes the whole cell be not pressurized at all, so it just collapses. And if you go through and make the soil, make the water hypertonic enough, you can make it where you can't grow plants for an extended period of time. So if you ever see that salt the earth phrase, it's true, and the whole thing they were doing was they were messing with osmosis by making it where the ground and the groundwater is simply too hypertonic for plants to survive, for plants to be hypertonic, which is what they need. Now, as we move on, we're going to kind of wrap up facilitated diffusion and get into active transport. So facilitated diffusion will oftentimes use these transmembrane channel proteins. And so these are just channels that let specific things, and I mean specific size, specific charge, sneak through. So you might have one that lets, for instance, like H pluses slide through it. And so while that channel is there, the H pluses will go from a concentration that is high in H plus to a concentration that is low. That's just kind of what they're going to do. And a lot of these too will have actual gates on them, so we'll oftentimes call them gated ion channels, because that's usually what we're working with facilitated diffusion is ions. And so we can actually close these channels to prevent this from happening. So if we build up a gradient, we can actually close this gate and let the gradient wait. And when we want to get rid of the gradient by opening up this channel, we can do so, and that will allow stuff to diffuse. Now, active transport, this is going to be the same basic idea where you use a protein, but the thing here is we're going to use pumps which are going to create a gradient. So they are going to expend ATP, and typically what happens is a phosphate will be attached to the protein, the specific transport protein, and that will allow a conformational change. It'll change shape. And so that one will allow it, by changing shape, to take something that's bonded on one side and change shape and move it to the other. And in some cases, they'll bind somebody on the other side then and bring somebody different back in. And so the whole process is going to cost us per flip, per change, 1 ATP. Because when we add the phosphate, we get 1 change, and when we remove it, we go back to the original state, which means we can then add another ATP and continue. So we'll have a variety of pumps that you'll see coming up here. We'll have H plus or proton pumps, and then we'll talk about on the next slide the sodium-potassium pump that are very common, necessary pumps for life to occur. Now, exocytosis and endocytosis, this will just be ways of dealing with large things in most cases. So when you see these, it's just large things either exiting or coming in. So exo, exit, endo, inside, you're bringing something in. So our poster child, our standard here for an active transport protein pump is going to be the sodium-potassium pump. This one is really cool. It's used by our nerve cells, our neurons. Our muscle cells use this to some degree. But this is going to be one that's known because it makes 3 different gradients with 1 pump. So it's pretty sweet. And what's going to happen here is, I always remember it, where your body, you don't want too much sodium. So the pump is going to actually pump 3 sodiums out. So 3 Na pluses are going to get pumped out of the cell. And so you can see in the picture here, you're expending an ATP to send 3 of them out into that extracellular space, that space outside the cell. You're then going to bring 2 potassiums in, right? People always need potassium, eat your oranges, eat your bananas. So we're going to bring 2 K pluses in. And so you can see here at the end that now that it's flipped shape, 2 potassiums bind, and then it removes that phosphate to let it go back to how it was. And so by going back to how it was, it shifts and brings those potassiums in, just like when it shifted the first time, it took the sodiums out. And now the ultimate result of this is we have more sodium outside the cell, so that's a gradient. The sodium would want to come in. We have more potassium inside the cell, which ultimately wants to go out now to get rid of the gradient. And because we brought 3 pluses in and we only let 2 pluses out, well, let me say this correctly, please. Because we brought 3 pluses out and we only brought 2 pluses in to fix that, we ultimately have this charge gradient where the outside of the cell is going to be positive and the inside of the cell is going to be negative. And so if you've watched The Matrix, where they say like humans are like batteries, what they're referring to is that our cells have what's called a membrane potential, where the inside of our cell is about negative 70 millivolts charged relative to the outside. And that's because of this negative charge that's built up inside because we pumped out extra positives. You know, too much positives outside. So you have 3 separate gradients that are here that all want to equal out. You know, positives want to come in. If there's negatives present on the inside, they would want to go out, as well as the sodium and potassium want to equalize. So this allows our cells to have a lot of stuff to play with when they're messing around with gradients, because one pump has given them 3 separate things to kind of tinker with. Now, as we wrap up things here, we've got exocytosis. What I really want you guys to get is that when you make a vesicle, usually from your Golgi apparatus, whatever you embed in the membrane of that vesicle is going to go up and it's going to merge with the plasma membrane. And so if I kind of try to draw it out, probably badly, as this vesicle reaches the edge of the cell, it's going to merge and kind of bubble into it, if you will. So it's going to create a little gap as it merges, so that there's a hole where you can essentially go from the outside of the cell into the vesicle. And then eventually it just kind of straightens out, and so we're left with just one solid piece of plasma membrane. This actually allows a cell to grow, because by adding vesicle membranes to our plasma membrane, the cell is now slightly larger. And this means anything now that we embedded, any enzymes, any pigments, any regular proteins that are structural, any carbohydrates, anything we embedded in that vesicle membrane is now part of our plasma membrane, or our cell membrane if you prefer. Anything inside the membrane has been released from the cell. It's excreted or secreted, and so this is like we talked about with the feces in the bag, where you can ultimately have your hand in the bag that's clean, grab it and flip, and now what's inside is outside the bag, and what was outside is inside the bag. So in this case, what was inside the vesicle is now going to be released to outside the cell. You can do this for a variety of purposes then, as I said, cell growth, to get stuff into our plasma membrane, as well as just to get large molecules that can't pass through either a protein, facilitated diffusion, or pump, or pass through the phospholipid bilayer itself. It allows us to get those things out. So if I ask you questions about like a large particle and what we're going to do, exocytosis is how we get rid of it. The opposite of this is going to be endocytosis, and endocytosis will be how we get big stuff typically into the cell. So it's going to be the same type process, except in this case we're going to kind of wrap around the object. So the plasma membrane is going to kind of fold up around what you want, and then once it touches, once it kind of wraps around enough that the sides touch, it'll pinch apart a vesicle. So this will shrink the plasma membrane slightly, because that vesicle membrane, or the food vacuole membrane, or whatever you pinched off, is membrane that used to be part of our actual plasma membrane, our cell membrane. Now there's 3 kinds of things that you can eat. Phagocytosis, or cell eating, is when you're actually taking in like a bacteria, or you're taking in like a large food particle. And so once you do that, you'll get a food vacuole when you're done, which you can typically merge a lysosome with. So we could have a little lysosome, or I suppose a peroxisome in some cases, could merge with that to digest it, so that would allow amoeba or a white blood cell to eat stuff. If you want fluids, you can do penocytosis, so that's a way of kind of, if you're doing any type of fluid homeostasis or fluid that you need to ingest because there's stuff dissolved in it, you could go through and do cell drinking, which would be penocytosis. This is the one you'll probably see least though. And then lastly, there's receptor-mediated endocytosis, where you want a specific particle, and you want lots of it. So what you do is you get these proteins that are receptors and grab hold of that particular, we call them a ligand, whatever we're after, and hold on to it. And so you put them in one specific region of your plasma membrane, and then you fold up and bring that region in, and because those particular receptors are holding on to that particular ligand, we can now kind of make sure we get the molecules we want. Otherwise, if you just kind of randomly grab, there's a good chance that you're not going to get the molecule you need. You're just going to waste energy. You're just going to use up a lot of membrane, bringing it in as a vesicle or a vacuole for no purpose. And so this one is a very, very specific, very targeted way of getting stuff into the cell versus some of the more general ways with phagocytosis and penocytosis, where they're not near as targeted for actual molecules, for relatively smaller things. And so that's our third type.